the real key to understanding chromosomes came with the understanding that DNA was the hereditary material. This story has been told many times, but just very briefly, it was found by studies on bacteria, by studies on bacteriophage, those are viruses that infect bacteria, and by studies on higher organisms, that the DNA was really the hereditary material. And all of that was really brought to a head with the experiments of Watson and Crick in the early 1960s, who showed what the structure of the DNA molecule is, and they showed how that structure would permit the hereditary material both to replicate itself and to serve as a template for the formation of proteins. And so by the 1960s, we understood that DNA was really the essential part of the chromosome. So a lot of my work during the 1960s and late 50s, for that matter, was devoted to trying to understand how the DNA was organized in the chromosome. The conclusion that we came to, my laboratory and others as well, was that the DNA was organized as a single long double helix from one end of the chromosome to the other. But there was a serious problem once we had discovered that the chromosome was one long piece of DNA. And that problem was that organisms, different organisms, have very different amounts of DNA in their basic makeup. Some organisms have as much as 10 times as much DNA as others, and yet they are organisms of the same general level of complexity, the same evolutionary scale. For instance, a grasshopper has roughly 10 to 15 times as much DNA as the fruit fly. A salamander has 15 times as much DNA as a human. And a human has 15 times as much DNA as some fish. Why do different organisms have different amounts of DNA when they all, one would think, would have about the same number of genes? So many people call this junk DNA, the DNA between the genes. So a lot of what we were interested in these in the mid-60s was the question of how the overall organization of the chromosome, or how the chromosome was built as a structure. And we knew that there were differentiations along the chromosome. We knew that there were regions called the centromere, which are the part of the chromosome that actually is pulled towards the poles when the chromosomes divide during cell division, during mitosis. We also knew there were big chunks of DNA around the centromeres that did not have genes in them. We knew that from genetics. So we knew there were large pieces of the DNA that didn't have genes in it. And one of the problems that my laboratory investigated was the question of the structure of this non-genic DNA.